Hello everyone, welcome back to part 3 of photosynthesis. Today we will be starting with the series of experiments, starting with the first experiment. So, for all the experiment of photosynthesis, the plant taken is a destarch one. So, what is the meaning of a destarch plant? A plant without starch is called a destarch plant. Now my next question, how will you make a plant destarch? How will you remove all the starch from the plant? So the answer is, if plant performs photosynthesis, what is produced? Glucose. And that glucose is stored in the form of starch. So for starch to be present, plant should carry out which process? Photosynthesis. So if photosynthesis occurs, starch is present. No photosynthesis means no starch. So how will you destarch a plant? By stopping the plant from carrying out which process? Photosynthesis. How will you stop the plant from carrying out photosynthesis? By keeping the plant in dark for 24 to 48 hours. Because for the process of photosynthesis, sunlight is very important. Plant cannot carry out photosynthesis in dark. So if you have to destarch a plant, if you have to remove all the starch from a plant, what you have to do? You have to stop photosynthesis. And how will you stop photosynthesis? By keeping the plant in dark for 24 to 48 hours. Now, how will you test whether starch is present in a plant or starch is absent? So, first to check the presence of starch in a plant, the test which is performed is iodine test. So, if you have to check कि plant में starch present है या absent है, कौन सा test करते हैं? आयोडीन टेस्ट करते हैं. तो आयोडीन की प्रेजेंस में अगर प्लांट ने ब्लू ब्लैक कलर दिया, इट मींस स्टार्च इज प्रेजेंट. आयोडीन की प्रेजेंस में इफ प्लांट गिव्स ब्राउन कलर, इट इंडिकेट्स स्टार्च इज एब्सेंट. सो अ क्विक रिकैप फॉर ऑल द experiments of photosynthesis, the plant taken is a destarch one. Why? Towards the end of the experiments I'll explain it. But how will you destarch a plant? Destarch matlab all the starch is removed. Kaise? How will you destarch the plant? For starch to be present, plant should carry out photosynthesis. So to remove starch, you should stop the plant from carrying out photosynthesis. How will you stop the plant from carrying out photosynthesis? By keeping it in dark for 24 to 48 hours. Or there is a test to check the presence or absence of starch in plant. Which test? Iodine test. Iodine की presence में if the plant gives blue black color, starch is present in the plant. Iodine की presence में if the plant gives brown color, starch is absent. Now coming to first experiment of photosynthesis, to test a leaf for starch. Starch का presence या absence test करने के लिए we do iodine test. And just like I told you, iodine की presence में blue black color indicates presence of starch and brown color indicates absence of starch. So what is done in this experiment? Let's understand the experimental setup. A beaker full of water. In the beaker there is a test tube. Test tube mein they have taken a alcohol. Which alcohol? Methylated spirit. Now this is the flame. So what is the procedure for the experiment? First take a leaf and dip the leaf in boiling water. Why do you think leaf is dipped in boiling water? To kill the cells. What is the meaning of to kill the cells? Guys, you remember 9th grade से कि cell membrane is semi-permeable membrane. Semi-permeable means selectively permeable. Cell membrane does not let any substance to exit or enter into the cell. It is very selective in nature. Only those substances will enter into the cell which are actually useful for the cell. So by keeping the leaf in boiling water or by dipping the leaf in boiling water, we are disrupting the cell membrane so that what can enter into the cell? Iodine. Otherwise, the semi-permeable nature of cell membrane will not allow iodine to enter into the cell. So it is very important to disrupt the cell membrane so that iodine can enter into the cell for starch test. So in the procedure, the first step is dip the leaf in boiling water to kill the cells. What is the meaning of to kill the cells? They are disrupting the cell membrane. Now the second step, boil the leaf in methylated spirit. What is methylated spirit? It is a alcohol. Boil the leaf in alcohol over a water bath. So the leaf is boiled in the alcohol but not directly on the flame. It is kept in a water bath. Why do you think this leaf in alcohol is not directly kept on the flame? Why are we boiling the leaf in alcohol in a water bath? Why in a water bath? Because alcohol is highly flammable in nature. If we will keep it directly on the flame, it will catch fire. That is why in the second step, leaf is boiled in the alcohol in methylated spirit, but it is done over a water bath. Now, why do you think leaf is being boiled in methylated spirit or alcohol? It will remove all the chlorophyll. Why do you think it is important to remove chlorophyll by boiling it in alcohol? Because the green color of chlorophyll can interfere with blue-black or brown color of iodine. That is why it's important to remove the green color of chlorophyll. So how do you remove chlorophyll? By boiling the leaf in methylated spirit over a water bath. Then take out the leaf and do the iodine test. Iodine की presence में if the color of the leaf turns blue-black, starch is present. If the color of the leaf turns brown, it shows starch is absent. Now, the next experiment in photosynthesis is to show that chlorophyll is important for the process of photosynthesis. Now, everyone knows what is the function of chlorophyll? It traps sunlight for the process of photosynthesis. No chlorophyll, no photosynthesis. So, what they have done in the experiment? They have taken a plant with variegated leaves. What is the meaning of variegated leaf? Variegated leaf is not entirely green in color. It has patches of green on it. So this is the diagram for a variegated leaf. It's not entirely green. It has patches of green and non-green parts. Which part of the leaf will carry out photosynthesis in the presence of sunlight? The green part or the non-green part? Obviously the green part, because the green part contains chlorophyll and chlorophyll is the one which will trap sunlight. So the green part of the leaf will carry out photosynthesis. If photosynthesis is happening, starch will be present. The non-green part of the leaf will not carry out photosynthesis because it does not have chlorophyll to trap sunlight. No photosynthesis means no starch. Now, if you will perform iodine test on this leaf, which part of the leaf will turn blue-black, which part will turn brown? The green part of the leaf will turn blue-black in presence of iodine, indicating that photosynthesis is happening in the green part. The non-green part will turn brown in presence of iodine, concluding that chlorophyll is very important for the process of photosynthesis. In your textbook, there are 3 examples given for the plants which have variegated leaves: coleus, croton and geranium. Memorize the names of the plants with variegated leaves because you can get an odd one out type of question. I'll give you one of the example. The question is, choose the odd one out and write the category to which others belong. And the question says coleus, croton, geranium and let's say an aquatic plant, hydrilla. So the question is coleus, croton, geranium, hydrilla. Choose the odd one out. Hydrilla becomes the odd one out. What's the category for the other 3? The plants with variegated leaves. Now the next experiment is to show that sunlight is necessary for the process of photosynthesis. So what is done in this experiment? They have taken a leaf from a destarched plant. Some part of this leaf is covered by black paper. So the part of the leaf which is covered by black paper is not receiving the sunlight. The other part of the leaf exposed to sunlight. Which part will carry out photosynthesis? The covered part or the exposed part? Obviously the exposed part will carry out photosynthesis. Starch will be present in which part? The covered part or the exposed part? The exposed part will carry out photosynthesis and starch will be present in the exposed part. Starch will be absent in the part of the leaf which is covered by black paper and is not receiving any sunlight. Which part will turn blue-black in presence of iodine? This or the covered part? The exposed part turns blue-black in the presence of iodine, indicating it has undergone which process? Photosynthesis. And what is present in it? Starch. The covered part of the leaf turns brown in presence of iodine, indicating it has not undergone the process of photosynthesis and it does not have any starch. The next experiment is to show that carbon dioxide is necessary for photosynthesis. You remember the reaction for photosynthesis? Carbon dioxide taken from atmosphere, water from the soil, in the presence of sunlight and chlorophyll. What were the products in photosynthesis? Glucose, oxygen and water. Can photosynthesis take place without carbon dioxide? No. Carbon dioxide is a very important reactant and without carbon dioxide plant cannot undergo the process of photosynthesis. So what is done in the experiment to prove this aim? They have taken a beaker. In the beaker a chemical is taken. What is name of the chemical? Potassium hydroxide. What's the formula? KOH. Then they have taken a destarch plant. Half of the leaf is inserted in the beaker which has a chemical KOH. The other half of part of the leaf is exposed to the air. Now what is the nature for potassium hydroxide? Potassium hydroxide has tendency to absorb carbon dioxide from its surroundings. So this potassium hydroxide will absorb all the carbon dioxide from the beaker. So in the beaker there is no carbon dioxide present. So half part of the leaf is in the beaker which does not have any carbon dioxide because this chemical KOH absorbs all the carbon dioxide from its surroundings. The other part of the leaf is exposed to carbon dioxide, is in the air, is getting carbon dioxide. Which part of the leaf will carry out photosynthesis? The one which is getting carbon dioxide or the one which is in the beaker and not receiving carbon dioxide? Obviously, the part which is exposed to the air will carry out photosynthesis. If it is carrying out photosynthesis, starch will be present or absent? Starch present. The part of the leaf which is in the beaker will not carry out photosynthesis, so no starch in it. If you will carry out iodine test on this leaf, which part of the leaf will turn blue-black in presence of iodine? The exposed one or in the beaker one? The part of the leaf which is exposed to air will turn blue-black in presence of iodine. The part of the leaf which is in the beaker will turn brown in presence of iodine, indicating that carbon dioxide is very important for the process of photosynthesis. They can give you the diagram, ask you to write down the aim for the experiment, name the chemical which is used in the experiment. So which chemical is used? Potassium hydroxide. What is the formula for potassium hydroxide? KOH. And what is the nature for potassium hydroxide? Potassium hydroxide absorbs all the carbon dioxide from its surrounding. Coming to the next experiment, to show that oxygen is produced during the process of photosynthesis. What is the experimental setup? Let's understand the experimental setup first. They have taken a beaker full of water. The plant taken in the beaker is an aquatic plant. The example given in your textbook for aquatic plant, Hydrilla, and the other one is Elodia. Memorize the name for the aquatic plants given. They can ask you odd one out type of questions. So the experimental setup, a beaker full of water, aquatic plant, either Hydrilla or Elodia. Now on top of aquatic plant, they have inverted a funnel. Can you see this funnel? On top of aquatic plant, a funnel is inverted. Now a test tube full of water, and that test tube is slowly inverted on the stem of the funnel. The entire setup is kept in sunlight for the process of photosynthesis. Now after few hours, what will be the observation? There will be bubbles seen in the test tube, indicating that some gas is being released by photosynthesis. How do we make sure that these bubbles produced during photosynthesis is of oxygen gas. So what is done? A glowing splinter is brought near the experimental setup and it bursts into flame, proving that these bubbles are of oxygen gas. There is one very important precaution for this experimental setup, which says the level of water in the beaker should be more than the stem of the funnel. I will repeat it. The level of water in the beaker should be more than the stem of the funnel, which basically means that the stem of this inverted funnel should be completely immersed in water. Why do you think it should be completely immersed in water? To prevent the entry of outside gases into the experimental setup. What will happen if the level of water in the beaker is not more than the stem of the inverted funnel? The outside gases can enter into the experimental setup and can contaminate the gas which is being collected in the test tube. So to prevent this, make sure the stem of the inverted funnel is completely immersed in the water. This diagram comes for drawing in your board exam for 3 marks. While drawing the diagram, make sure this important precaution is followed. This is very, very important. If you do not follow this precaution while drawing the diagram, you are going to lose your 3 marks. So be very, very cautious while drawing the diagram about this precaution. So before starting the next topic, I would like to conclude about all the experiments of photosynthesis. There is one very important application based question. So we have seen series of experiments for photosynthesis. In all the experiment for photosynthesis, there was one common thing. सारे experiment में the plant taken at the start of the experiment was a destarch plant. Now my question is, why do you think in all the experiments of photosynthesis they have taken a destarch plant? See, what is the answer? Start of the experiment, end of the experiment. If you will take a destarch plant at the start of the experiment, कि start of the experiment में तो starch नहीं है plant में, then you keep the plant in sunlight for 2-3 hours. Or after 2-3 hours, the plant shows the presence of starch. Then you can conclude that the plant has undergone the process of photosynthesis. But अगर start of the experiment में भी starch present है, then you keep it in sunlight so that it can undergo the process of photosynthesis. And after 2-3 hours towards the end of the experiment भी starch present है. How will you conclude that plant has undergone photosynthesis? That is why start of the experiment में destarch plant लिया कि starch नहीं था when we started the experiment. Sunlight में रखा, photosynthesis हुआ, after 2-3 hours towards the end of the experiment starch present है, and we can conclude that the plant has undergone the process of photosynthesis. That is why the starch is being formed. Is this clear? Now moving to the next topic, which is food chain. I am sure everyone knows what a food chain is. So in simple language, food chain basically tells you who eats whom in nature. Nature में किसने किसको खाया? This is what you get to know from food chain. I'll give you a simple example. In the nature, grass is eaten by grasshopper. Grasshopper is eaten by frog. Frog in turn eaten by snake. In the end, snake is eaten by eagle. So who eats whom? किसने किसको खाया? And the food chain always starts with producers. Food chain always starts with producers. What are producers? Plants are called producers because they can produce their own food. By which process? By photosynthesis. So food chain always will start with producers. What are producers? Plants. And why it always starts with producers? Because producers can make their own food by the process of photosynthesis. And this is how food or energy is being passed on from one organism to another in nature. A question, name the following question, which was actually one of the board paper question. Name the physiological process which starts the food chain. So what will be the physiological process which is starting the food chain? Photosynthesis. What is the answer? Photosynthesis. Photosynthesis is the physiological process which will start the food chain. So this is producer, primary consumer, secondary consumer, tertiary consumer and the top consumer. They can ask you arrange them in logical sequence in your board paper. So there are multiple examples given for food chain in your textbook. Just go through all the examples once. The question which can come from these examples of food chain is arrange them in logical sequence in section A of your paper. So that's it for photosynthesis chapter. Let's meet in the next chapter. Bye.